Okay, very good morning, my dear friends. So welcome, Dr. A. Rajini Madam Garu, Head Department of Botany, Food and Nutrition, RBVR Women's College. So this is Dr. Padala Thirupathi, Assistant Professor. So in the Faculty Exchange Program, we are here. So today we are going to discuss. Madam, may I continue?
Okay, sir. Yes, sir.
Yeah. So today we are going to discuss about the one of the important area in the microbiology. In previous class, we have already discussed about the archaebacteria and the reproduction in bacteria and nutrition in bacteria and other things we have clearly discussed. But today itself, we are going to discuss about the another topic in microbiology, that is Actinomycetes, my dear friends. So today, Actinomycetes, we are going to discuss here. The slide is visible?
No, sir.
Now?
Yes. Okay, sir.
Right. So, this is Dr. Padala Thirupathi, Assistant Professor, Department of Botany, Women's Degree College, Hyderabad, Women's Degree College, Jagityal. So, Rajini Madam Garu.
Cheppandi.
So, Women's Degree College, Jagityal. So, today's topic, Actinomycetes. In this Actinomycetes, we are going to discuss the content of today's class. That is the history about the Actinomycetes and the classification of the Actinomycetes. Okay. Okay. A classification of the Actinomycetes and introduction, morphology, a role of Actinomycetes, isolation, and different sources and media for the isolation of this archaebacteria, and what are the applications related to production from this Actinomycetes of the different enzymes and the antibiotics. Okay, and nitrogen fixing bacteria, that is including Frankia also, and decomposer of Actinomycetes. Now, the history about the Actinomycetes in 1940s, no one had really though much about the bacteria of Actinomycetes. So that is until Selman Waxman discovered streptomycin, the first treatment of tuberculosis that actually worked. So this streptomyces belongs to Actinomycetes. So Actinomycetes means the bacteria which they are related, similar fungal member, fungus-like bacteria. So fungus-like bacteria we can call as Actinomycetes bacteria. After discovery of this, the streptomycin first used by Selman Waxman for the treatment of the tuberculosis. Then after the 1945, many different types of the bacteria, they are related to fungal members that we can call as Actinomycetes bacteria. Then they are used for the extraction of the number of antibiotics. The first antibiotic, we know that first antibiotic is penicillin, that is extracted from fungal member. But the other antibiotics like streptomycin, kanamycin, terramycin, gentamicin, these are all antibiotics which are extracted from bacteria. So these bacteria belongs to Actinomycetes bacteria. In 1940, Selman Abraham Waxman, with the help of Bayard Woodruff, discovered the antibiotic that is actinomycin. So actinomycin is mostly made up of the bacteria Actinomycetes. Actinomycin, it is an antibiotic. It is an antibiotic which are extracted from the Actinomycetes bacteria. Well, while the preliminary antibiotic failed, that it led to other varieties such as actinomycin A, actinomycin B, and actinomycin C and D also. And several these new varieties were found to have strong anti-cancer effects. These antibiotics, they are used as anti-cancer effects for the treatment of the cancer. And after about the 50 years later, so the biotechnology and also the genetic research on Actinomycetes now are used in most of the antibiotics used to treat infections and drugs that are anti-cancer, anti-parasitic, and immunosuppressant. So these are the development of the discovery of the antibiotics by the involvement of the genetic research and also the advanced development of the biotechnological research. And the Actinomycetes, am I audible? Am I audible, my dear friends?
Yes, sir.
So
Student, tell me.
these Actinomycetes group of the bacteria, the Actinomycetes bacteria, they are belongs to gram-positive bacteria. The order of these Actinomycetes are Actinomycetes that produce various bioactive agents, including antibiotics, enzymes, and vitamins also. So the major producing antibiotics, they are belongs to Streptomyces bacteria. And the classification of these Actinomycetes is they are belongs to order Actinomycetes under the families, that is the Mycobacteriaceae, Actinomycetes, Streptomycetes, and Actinoplanaceae. These are the four families belongs to Actinomycetes order. Am I clear? How many orders we are discussed in this class, in this slide? This slide is visible. How many order is here? How many orders are here? Only one order, that is Actinomycetes. How many classes are here? Sorry, how many families are here? Mycobacteriaceae, Actinomycetes, Streptomycetes, and Actinoplanaceae. How many families are here, my dear friends? 4 families.
One family.
4 families. So this Actinomycetes genera, which are agriculturally and industrially very important source. Okay. So only 2 families of this Actinomycetes order, that is Actinomycetes and Streptomycetes, are more useful. In the order of abundance in soils, the common genera of the Actinomycetes are Streptomyces. Nearly 70% of the Actinomycetes bacteria which they are present in the soil. And the best example of this bacteria, of the Streptomyces, Actinomycetes bacteria, they are Nocardia and Micromonospora. Micromonospora. So the gram-positive bacteria, the Streptomyces bacteria are the gram-positive bacteria. They are the bacilli. The cell wall and internal structures are typical of the bacteria rather than fungi. So the cell wall and other internal structure very similar to the bacteria. We know that the cell wall is made up of murin or mucopolypeptide or peptidoglycan, and the plasma membrane is also made up of a lipoprotein layer. Okay. Yes. Any doubts? Any doubt, Shivani? Any doubt? Are you discussing? D. Shivani. Okay. So these Actinomycetes, they are aerobic and anaerobic bacteria also. Aerobic means they need for the metabolic activities and other activities, oxygen is necessary. And the other Actinomycetes, they are anaerobic type of bacteria. Okay. But remember, my dear friends, these Actinomycetes bacteria they are growing very slowly compared to Archaebacteria and other bacteria. They are non-motile because they does not possess any motile organs like cilia and flagella. Okay, but including these bacteria, the Actinomycetes bacteria, they are non-sporing. They are not bearing spores. They are non-capsulated. So the cell structure compared to bacteria and Actinomycetes, they are non-capsulated bacteria. And the bacteria are the filamentous bacteria. They are filamentous bacteria. They may be the branched or they may be the unbranched filamentous bacteria. They are belongs to Actinomycetes bacteria. They are majority of the Actinomycetes bacteria. They are grown in the soil, and also they are grown the extreme conditions like our human intestine, in our alimentary canal also. And the important genera including of this Actinomycetes, they are Actinomyces, Nocardia, Actinomadura, Streptomyces, and Thermophilic. Okay, these are the genera which they are belongs to Actinomycetes. Actinomyces, Nocardia, Streptomyces, Actinomadura, and Thermophilic. Clear? So the common type of Actinomycetes bacteria, they are looks like here. They are looks like the filamentous, branched filamentous Actinobacteria we are observed in this slide. So one spore is going to germinate to produce hyphae, like fungal hyphae. They are septate. And they are in the developmental process, they are shows number of branches. Am I clear? And what are the functions of this Actinomycetes? What is the role of this Actinomycetes bacteria? Number one, they are the decomposers or degrading of the organic matter. They are depending upon the dead and decaying substances. Okay, like cellulose, polysaccharides, protein fats, and the organic acids. And some of the bacteria, they are organic residues. Some of the bacteria, they are organic residues. Okay. See here, they are the decomposers, and some of they are organic residues, substances added for the soil, and the first attacked by bacteria and fungi, and later Actinomycetes, because they are very slowly they are grown. The growth of these bacteria is compared to bacteria and fungi very slow manner. Okay, and the decomposed degrade more resistant and indecomposable organic substances, and they are produced number of dark black brown pigments which contribute to dark color of the soil humus. The dark color of the soil humus just because of the Actinomycetes bacteria. They are also responsible for subsequent further decomposition of the humus resistance material to material in soil. This is nothing but the continuation of the biogeochemical cycle for the added of the minerals in the soil. Okay, they are responsible for earthy, musty odor smell of the freshly plowed soils. Whenever the first rainfall has come, the odor and the smell has come from the soil just because of the Actinomycetes bacteria present in the soil. Am I right? Are you agree or not? Are you ever observed the smell comes out from the soil from the first rainfall? Are you here? Are you listening? Are you listen?
Yes, sir.
So just because of this Actinomycetes bacteria, the odor or the different smell has comes out from the soil whenever the first rainfall has occurred. And one of the species of Actinomycetes, that is the Streptomyces scabies, cause disease. We are observed in the plant, that is a potato scab in potato, by the Streptomyces scabies genera. Okay, Streptomyces scabies is responsible for the disease in the potato scab, disease in potato. And the how to isolation of this Actinomycetes bacteria from the various sources of the soil, or maybe the other other disease of the plants. So collection of the samples from the different habitats and enrichment of a sample using different treatments. Sample were cultured in different media and isolate by morphological, physiological, biochemical and molecular methods, and strain of Actinomycetes is isolated. These are the method of the whenever using the isolation of the Actinomycetes bacteria. And after gaining of the stains of the Actinomycetes isolated, then we are using morphological methods and physiological methods and biochemical methods and molecular methods also. We are applied for the isolation of the stains of the Actinomycetes bacteria. The molecular methods, they are RFLP method, or even though the RAPD, then PFG and ARDRA. These are the molecular methods, my dear friends. Molecular methods by the using of the molecular markers, RFLP, RAPD. These are the molecular markers for the separation of the Actinomycetes bacteria. And the biochemical methods also we can apply. They are catalase production, urease production, hydrogen sulfide production, nitrate reduction, starch hydrolysis, and gelatin liquefaction, and methyl red test. Okay, these are the tests we are using in the biochemical methods. And physiological methods, the pH, range of the pH, maybe the alkaline or the acidic nature, and the optimum temperature, room temperature 2 to that means 20 to 25 degrees Celsius. Okay, and the different temperature we can use. And salinity. Salinity means the value, pH value, more than 7 pH, the concentration of the hydrogen, that we can call as a salinic condition. And morphological methods, they are we are using macroscopic and microscopic methods and slide culture methods for the isolation of the Actinomycetes bacteria. And different source for the isolation of the Actinomycetes bacteria. The source may be the soil, the source may be the fast soil or humus layer, or even though the cornfield or cow barnyard, even the forest and lake, or even the lake soil, normal soil, even the marine soil. So these are the sources from the sources of the Actinomycetes bacteria. And even though water also the source of the collection of the Actinomycetes bacteria. So stream sediments and lake muds and marine sediments also the major source of the isolation of these Actinomycetes bacteria. And what are the applications of these enzymes production of this Actinomycetes bacteria? What are the enzymes are extracted from the Actinomycetes bacteria, and that enzymes used for which type of treatment or which type of benefits we are gaining from this Actinomycetes bacteria? See here, this is the amylase, is an enzyme which is extracted from the Actinomycetes bacteria. These amylases are the enzymes which are used for the in fermentation, in food industry, or even the textile and paper industry. Are you following, my dear friends?
Yes, sir.
The another one is the catalase enzyme. The catalase enzyme used for as an antioxidant in even though the cold sterilization of the beer in dairy industry, also catalase enzyme. And cellulases enzyme is useful for the in biomechanical pulping, in laundry and animal feed industry. And see here, the chitinase enzyme. This chitinase enzyme in plant resistance against fungal pathogen, in drug delivery and wound healing. In biochemical industry also this chitinase enzyme is useful. And protease enzyme, proteases, these protease enzymes used as anti-inflammation, protect against clots in cancer treatment also. Urease enzyme, my dear friends, remember the first synthetic enzyme which is produced inside the research laboratory. The enzyme is urease by Sumner. Urease enzyme first crystallized by Sumner, J.B. Sumner. J.B. Sumner was the scientist first crystallized enzyme. That enzyme is nothing but urease. This urease enzyme, which is used in the wine industry, beverage industry, and also used for the analysis of the blood and urine in the diagnostic centers, and analysis of the heavy metals content in wastewater and soil. Whenever we are catching or the urban areas, metropolitan areas like Hyderabad and Twin Cities and Delhi and other urban areas, the analysis of the treatment of the waste management, they are analyzing, the scientists, they are analyzing which type of metals are present in that garbage or wastewater. Okay, and lipase enzyme, my dear friends, remember this is the most important enzyme for the biochemical, especially the synthesis of the, or even the preparation of the what we can say that they are making of the detergent industries and diagnostic centers and pharmaceutical industries also, lipase enzyme. Okay, and L-aspargine enzyme. This L-aspargine enzyme, this is for the treatment of the bone marrow. Some of the members, they are some of the human beings and other members, they are affected by the cancer, especially the bone marrow or the bone cancer. They are treatment of the bone marrow and in stem cell transplantation also. Have you ever heard the term stem cells? So these stem cells transplantation also by the involvement of by the uses of the L-aspargase. L-aspargase is an enzyme which is used for the treatment of the bone marrow and also in stem cell transplantation also. And in treatment of the acute leukemia. Are you ever heard the term leukemia, my dear friends?
Yes, sir.
Leukemia is also nothing but the blood cancer, my dear friends. It is also we can call as Pandurogam. Okay, in Mahabharata, Panduraja has affected by the this disease that is nothing but leukemia. The excess production of the leukocytes. By the production of these leukocytes, the blood cells is extracted from our body. They are elicited from our nose and mouth and also ears in the leukemia cancer. Okay, so these are the enzymes which are produced from the Actinomycetes bacteria. In this regard, this slide, what we are observed, Amma? By the using of this Actinomycetes, number of enzymes are produced and they are used in different ways for the human welfare. Am I right or not? And the applications of the Actinomycetes. The Actinomycetes for the biocorrosion and the biopesticide agents, and also biomedical production of the biomedical products, and the production of the bioremediation, and using with the bioremediation, and the using of the biocontrol tools, and also applications in plant growth hormones for the production of the plant growth hormones, and also using plant growth promoting agents, and also agroactive production of the active or bioactive compounds, and biocorrosion. These are the all applications of the Actinomycetes bacteria, my dear friends. Are you following? Am I audible?
Yes, sir.
Yes, sir.
So in this slide, what you are observed, Amma? Actinome by the usage of the Actinomycetes. These are the applications in our day-to-day life activity. Horticulture, agriculture, industry, research. Am I right? Even though the bioremediation, biomedical, biopesticide agents, okay, for the production of the biopesticide agents also from the Actinomycetes bacteria. So Actinomycetes bacteria we can also use as a biopesticide agents. We are not using the pesticides, chemical pesticides. Whenever we are using excess usage of the chemical or the pesticides, they are shows about the abundant impact on human life and also the other environment factors. And by this reason, we are using of the Actinomycetes as a biopesticides in the replacement of the chemical pesticides. Whenever we are using of the chemical pesticides, the excess amount of the usage of the chemical pesticide, they are reached the nearest water bodies or ponds or lakes or even to the streams. In that aquatic fauna or flora, they can absorb these chemical pesticides, and also the chemical pesticides through biomagnification cycle, it reaches the human body. That effects of the most important the organs of the our body, especially the critical organs, they are affected by these chemical pesticides and chemical fertilizers also. In this regard, we are using in the place of chemical pesticides, Actinomycetes are used up as a biopesticide agents. Are you agree or not? Am I right or not?
Yes, sir.
Then what about the biocontrol tools? Biocontrol tools means the controlling agents. Whenever we are using the chemicals or other organic substances, the Actinomycetes itself used as a biocontrol tools. And also plant growth hormones. What are the plant growth hormones? Have you ever heard the term plant growth hormones?
Yes.
What are they?
Auxins, ethylene.
Only two hormones are you studied in your earlier classes?
Sir.
Only two are studied in your earlier classes?
Gibberellins, cytokinins, sir.
Very good, Amma. Yes, nice response. Gibberellins, cytokinins, auxins, abscisic acid, ethylene, and also some of the brasinosteroids. These are all plant growth treated as plant growth hormones, you know. So these plant growth hormones also, by the usage of these Actinomycetes, abundant production by the involvement of this Actinomycetes bacteria. And the plant growth promoting agents also by the production of the vitamin substances, other minerals also, by the continuation of the biogeochemical cycles. And the agroactive compounds. Agroactive compound means which they are promote the growth of the root. which they are promote the stem of the plant. So the plant growth is promoted by the involvement of this Actinomycetes, and also the texture of the soil, and also the whenever the Actinomycetes are applicable or application in the soil, whenever the treatment of this Actinomycetes bacteria, the soil is very useful manner, and the texture is changed for the easily growth of the root and the promoting of the stem, and also the health of the crop, or even though the plant is growing very healthy manner without using any chemical fertilizers. So this Actinomycetes is also as a agroactive compounds for the growth of the crops. Is it right or not? And the nitrogen fixing bacteria, Frankia, they are also belongs to these Actinomycetes. Are you ever heard of Frankia? Frankia is a bacteria. It is an Actinomycetes bacteria. So 75 in atmosphere, we know the gaseous substance molecular state of the nitrogen, which they are present in the 78% of the nitrogen is present. Are you agree or not? Come on, respond, my dear friends. Are you feel bored? Are you feel bored, my dear friends?
No, sir.
So 78, more than 78% of the nitrogen is present. Highest amount of the gaseous state of the gas which they are present in the atmosphere, that is nitrogen, you know. But my dear friends, remember this molecular state of the nitrogen, which they are all higher plants, they cannot use in this form. In this reason, this Frankia or Actinomycetes bacteria, they are involved fixation of the nitrogen from the atmosphere by the useful form. The useful forms, they are maybe the nitrates, maybe the nitrites, or ammonia or ammonium. These are the usable forms of the higher plants of the nitrogen. The natural nitrogen cycle, the nitrogen cycle, they are relies on the nitrogen fixing bacteria like those found of the Frankia family of the Actinobacteria. They are supply of the fixed nitrogen is often the limiting factor for the growth. So in this regard, the 15% of the world nitrogen fixed naturally is from symbiotic manner of this Frankia family Actinomycetes bacteria of their host plants. Okay, mainly ulnus plant. They are ulnus plants. They are having the root nodules by the involvement of this Frankia bacteria. This Frankia, which they are in the symbiotic manner, they are associated with the ulnus plants. These ulnus roots, they are producing root nodules by the involvement of this Frankia and also fixation of the nitrogen from the atmosphere. Okay. About the 15% of the world nitrogen fixation by the symbiotic manner by the Frankia family. Okay. And the plants that form symbiotic relationship Frankia are called actinorhizal plants. Which plants, Amma? Actinorhizal plants. The Frankia is able to provide most or all of the host plants of the nitrogen needs. Okay. So these, in this manner, the atmospheric nitrogen or molecular nitrogen is converted into usable forms of ammonia, nitrates, and nitrites by Frankia family or the Actinomycetes bacteria. So these are the root nodules we are observed in the ulnus roots. These root nodules, are you seen in this slide, Amma? So left image, this left image shows about the root nodules and the right image shows about the bacillus. Are you ever heard, Amma? Though our Actinomycetes bacteria, majority of the Actinomycetes bacteria, they are bacilli. Bacillus means rod-shaped bacteria. This is gram-positive bacteria, my dear friends. Am I right or not? Are you agree? Gram-positive rod-shaped bacteria. So these bacteria are involved for the production of the root nodules in the ulnus plant. These bacteria, nothing but Frankia family, Actinomycetes bacteria. They are producing root nodules in ulnus plants. These root nodules is able to fixation of the nitrogen from the atmosphere. Is it clear? And some of the Actinomycetes some of the bacteria, they are used as a decomposers. Means they are cleaning the earth's surface, dead and decaying material, and all the dead bodies of the plants and all animals, they are decomposed by the using or by the involvement of these Actinomycetes bacteria. And also they are added the minerals and amino acids or even the proteins and other substances by the involvement of these saprophytic nature bacteria. That nothing but decomposer Actinomycetes bacteria. That's why we can call as scavengers of the natural scavengers of the entire earth, Actinomycetes bacteria. And they are also treated as they are grown in very extreme temperature that we can call as thermophiles. That bacteria we can call as thermophiles. They are thermophilous. Philus means like. They are loving high temperature environment. Philus means like, love, affection. Okay. So they are most loving environment. The Actinomycetes bacteria, they can sustain, they can survive higher temperature just because of they are love of the heat environment, highest temperature. They can survive. They can survive in highest temperature. That's why these Actinomycetes bacteria, that type of Actinomycetes bacteria, they are, we can call it thermophiles. Okay. And that what we are observed, whenever we are going to burn the garbage, burn the waste in rural areas and outskirts of the city, in that temperature also this thermophilus Actinomycetes bacteria, they can survive. Am I right or not? More than 80 or 90 degrees Celsius. Are you agree or not, my dear friends? Respond.
Yes, sir.
So that's why the Actinomycetes bacteria we can call as the decomposers, and they are the natural scavengers of the entire earth, my dear friends. Okay. So this is about the Actinomycetes bacteria, my dear friends. Regarding of this class, if you have any doubt, please ask me, my dear friends. Within 1 minute, our class is going to wind up. If you have any doubt, please ask me. No doubt?
I think no doubt, sir.
Oh, you are very good student, because of your fresh mind. That's why I prefer take the class in morning. Your fresh mind is going to receive any information regarding of our class, even though new topic. Okay. Thank you.
Okay, sir. Thank you, sir.
Sir.